Chapter 6, the Muscular System, Screencast 2. Now, this screencast will be focusing on the actual contractile process, and this all occurs at the microscopic level. So we're now actually going smaller than the muscle fiber or muscle cell. Now, remember, skeletal muscle is also described as striated, and that is due to this particular structure, the sarcomere. The sarcomere is also the contractile unit of muscle. So this is actually at the microscopic level once again. And we did talk about two previous contractile proteins, and those are going to be myosin. And myosin actually is going to be responsible for the thick filament. And then we have actin, which is the thin filament. Now, we're also going to be looking at the general architecture of that sarcomere. And one of the things that we want to introduce is the Z line. This is the actual boundary of the sarcomere, and it's actually going to be the attachment point. Now, a sarcomere is a segment of myofibril extending from one Z line to the next, and that's why I use the term boundary of a sarcomere. Now, the arrangement of thick and thin filaments is actually what gives rise to the striated appearance of not just skeletal muscle, but also cardiac muscle. So based on the anatomy, it's safe to say that cardiac muscle is going to function very similar to skeletal muscle. But keep in mind one really critical difference. Skeletal muscle is voluntary, while as cardiac muscle is not. Very, very important distinction to keep in mind, even though the anatomy of the two is almost identical at the microscopic and functional level. Here we can actually see a myofibril, and notice what we have here. We actually have a Z line to Z line, and that's actually the boundary of a particular sarcomere. So let's take a closer look, and what we're going to see here, there's the actual Z line. Here are the thin filaments, and here are the thick filaments. Now, what we're going to encounter a little bit later is how these two filaments actually interact and form what we are going to call a cross bridge. So something to keep in mind. Here we can actually look at a different view. This is a cross section of a sarcomere, and we can actually see the arrangements of the thick versus the thin filament. And here we're also kind of looking at a sarcomere. So here's the Z lines here, and the gap once again from here to here is the actual sarcomere. Now, when we look at what's happening with the sarcomere, this is an important thing to keep in mind. Each sarcomere will actually shorten a little bit. That's the actual process of contraction. Now, what we're going to see here, the skeletal muscle is what we refer to as being neurogenic. So it has to be activated by a motor neuron. And what we're going to see here is what that nerve activation actually does. It's going to increase the availability of calcium around those contractile proteins, actin and myosin. Now, the presence of calcium is going to allow contraction to take place, and when that nerve is no longer stimulating the muscle, contraction will then stop. Now, motor neurons, as indicated, those are the ones that are actually responsible for stimulating muscle contraction. Now, the neurotransmitter that's going to be involved in this process is acetylcholine, and that is going to be released from a motor neuron at a neuromuscular junction. Those electrical impulses are going to be transmitted along what we're going to call T-tubules. Now, within, there's going to be a structure we refer to as the sarcoplasmic reticulum, and in skeletal muscle, let's think of that as the calcium warehouse. And that's what's going to happen when the electrical impulse is transmitted. This calcium is going to be released, and then calcium is going to be responsible for actually initiating a chain of events that are actually responsible for contraction when it interacts with the myofibrils. Here, let's take a look. There's our motor neuron. Okay, acetylcholine is going to be released. Here, this is that neuromuscular junction. So this is a chemical synapse. We saw these previously. So this release of acetylcholine is actually going to result in an action potential to be generated inside the muscle cell plasma membrane. This electrical impulse. So here's our T tubules, okay? So this is going to be carried to the cells, and around here, this whole blue structure, this is the endoplasmic reticulum or the calcium warehouse. And so what this electrical impulse is going to be responsible for eventually is to trigger the release of calcium. And notice what we have here. We basically can see all of these myofibrils. Now, the thick filaments are myosin, the thin filaments are actin, and what we're going to see is actually a cross bridge between the thin and thick filaments. I alluded to this in a previous diagram. Now, the actual contractile process is due to the formation of cross bridges between thin and thick filaments. Now, calcium is necessary for cross bridges to form. In the absence of calcium, there's going to be a protein complex referred to troponin and tropomyosin. And what this does, this actually covers the binding site for myosin on actin molecules. In the presence of calcium, this binding site will be available and a cross bridge can be formed. Now, what we're looking at is the initiation of what's referred to as the sliding filament mechanism, and calcium is released from the sarcoplasmic reticulum, and we'll see a diagram of this in just a little bit. Calcium will bind to troponin. The troponin-tropomyosin complex will change position. Now, the myosin binding site is actually exposed. The myosin head will actually connect with the actin, forming a cross bridge, and actin filaments are going to be pulled towards the center of the sarcomere, resulting in the actual shortening of the sarcomere. Myofilaments do not shorten. It is the sarcomere that actually will shorten. Now let's kind of take a look at this overall. So here we can see the thick filament, the thin filament, okay? And really what we're looking at here is the relaxed state, okay? So notice there's no cross bridges here. Here we can actually see the myosin head, but there is actually the actin molecule there. So somehow we have to create an interaction. Now, what we're going to see here during the contractile process, now we see the formation of cross bridges between the myosin head and the binding site on the thin filament made up of actin. And what's going to happen here is actin will actually, they'll bend, and what we're going to see here is the movement of the actin filament closer. Now, when nerve activation actually ends, contraction will end. Calcium will be actively pumped back into the sarcoplasmic reticulum, and that's going to require ATP. Now that calcium is no longer bound to troponin, the myosin binding site is covered, and the ATP must bind to myosin before myosin heads can detach from actin. No calcium, no cross bridges, muscle actually relaxes. So here, a couple of things. There's our thick filament, and this green here, okay, we can actually see the tropomyosin, and that's actually covering. So the green line here is the tropomyosin, and these here, this is actually the troponin. Now notice what happens. Calcium will actually bind to the troponin. Once that occurs, that kind of pulls the tropomyosin away, and we can now form a cross bridge. So that's how the actual connection between the myosin head and the binding site on actin will actually take place. Now, this is going to require ATP. ATP is going to be required for the actual contraction. It's required for the relaxation. Now, there's a number of mechanisms in which ATP is actually going to be replenished. Now, this is happening all the time. We have creatine phosphate, and that's actually a phosphate reservoir. We have glycogen that's stored within muscle and the liver. And the primary mechanism of generating ATP is the aerobic metabolism of glucose, fatty acids, and other high energy molecules. Now, here we can look at the sources, so stored ATP, very small quantities, and we basically use all of that in about 10 seconds. Now, remember, ATP is the only direct energy source, and we're going to regenerate that from other molecules. Now, stored creatine phosphate, let's think of this as a phosphate reservoir, and we have about 3 to 5 times amount of creatine phosphate than we do stored ATP. This will actually last for about 30 seconds. Now, creatine phosphate is very quickly converted into ATP. Then we have stored glycogen, and this is variable. It's going to be dependent upon muscle stores, and different muscles are going to have different stores. This is primarily used during heavy exercise within the first 3 to 5 minutes. Now, the ATP yield depends on whether or not oxygen is available. One glucose molecule derived from stored glycogen is only going to give us about 2 ATP molecules in the absence of oxygen. But if oxygen is available, we'll actually get up to 36. Then we have the aerobic metabolism. This has a steady high yield. Oxygen and nutrients, glucose, fatty acids that are derived from fat, are constantly supplied by the blood. Now, this is always present. It increases dramatically within several minutes of the onset of exercise, with blood flow and respiration increases. The complete metabolism of one glucose molecule will yield 36 ATP molecules. So aerobic metabolism is the primary way in which the body replenishes ATP stores to fuel muscle contraction.